So a warm welcome to one and all present here. Today we are going to see the topic immunochemistry. So as immunoglobulin has the another name which is called as antibody, and this immunoglobulin is mainly comes under the immunochemistry, and this immunoglobulin is also called as antibody. So these antibodies are normally present in our body, and its structure, it includes a very large Y-shaped molecule, and it is mainly produced by the plasma cells, and they bind to foreign particles and invade them, get attacked by the antibody. So one set of antibodies there, another antigens are there, which is the foreign pathogens that invade the body, and this antigen have the capability to give rise to a response from our immune system by producing the antibodies. So hence antigens stimulate the production of antibodies by the immune system. And the immunoglobulin is also called as antibodies, and these are glycoprotein molecules produced only from the plasma cells or white blood cells, and they specifically recognize and bind to particular antigens which attacks our body. And coming to the structure, so this, as I have told already, this antibody has Y-shaped structure, and it is made up of mainly 4 polypeptide subunits, and each subunit has 2 identical light and heavy chains, and it is also having the N-terminus sites of each heavy chain, thus forming an antigen binding domain with the light chain. So these structure has 2 light chain and 2 heavy chains, and these are 2 antigen binding domains forming the arms of the Y shape, and this is commonly termed as fragment antigen binding domains. And the C terminus of the heavy chains forms fragment crystallization, and which mainly helps in the interaction with the effector cells. And all 4 polypeptide subunits are held together by one bond, which is called as disulfide bond and non-covalent bond. So each antibody has 2 identical antigen binding sites, one in left side and another one is right side, and they differ in the antibody binding. So coming to the structure, these are the common Y-shaped structure. As we told already, it has a H chain, that is H heavy chain, and L light chain, and left side we have seen the NH3 terminus, that is N terminus, and another side is C terminus, that is carboxyl terminus. And if we have found the fragment antigen binding and fragment crystallization binding is there, where C is the constant domain and V is the variable domain, and H and L is the heavy chain and light chains. And we have seen the antigen binding site just in the top of the antibody. So in antigen binding site is the one in which the antigen gets bind and interacts with the antibody and forming the reaction. And coming to the classes, we have 5 subclasses of immunoglobulin, that is IgG, IgM, IgA, IgD and IgE, where IgG is gamma and M represent mu, and A represent alpha, D represent delta and E represent epsilon. And coming to the subclasses of gamma and alpha, we have found 4 subclasses in IgG, that is gamma 1 heavy chain, gamma 2 heavy chain, gamma 3 heavy chain, gamma 4 heavy chain, and IgA subclasses is regarded as alpha 1 heavy chain and alpha 2 heavy chains. So coming to the types of the antibodies, you have seen the liberal explanation of IgM. So IgM, it is immunoglobulin mu molecule. It is the first antibody which is produced in response to the microbial attack by the B lymphocytes, and it is termed as the largest antibody, and it is found in the pentameric form. So it has 5 different forms in their structure, and it circulates in the blood and lymph nodes, and it constitutes about 6% of the total antibody content, and it is mainly involved in the agglutination and opsonization reaction, and it is also having the large number of antigenic sites, so in which the surface in which the antigen gets bind, and therefore facilitates the efficient activation of the immune system. So this IgM is regarded as the first and largest antibody which is found inside our blood, and it is mainly responsible for the attacking of microbial pathogen by T cells.